Video we're going to be talking about something called gene regulation. And now both prokaryotes and eukaryotes do this, but they do it differently. So that's kind of where we're going to start. I'm going to review you on the difference between prokaryotes and eukaryotes again, and set up the idea that these two need to be regulating their genes in a different way. So first, here's our little picture. Now we remember that prokaryotes, are single-celled organisms. They do not come together to form a larger organism. They exist individually as an individual organism, and they're very simple. They simply have a little loop of DNA. They do not put it inside a nucleus, and they have ribosomes and maybe some vacuoles here to store some water or something like that. They might have a flagella, and they have an outer membrane. And then, of course, they are filled with cytoplasm, whereas where most things happen inside the prokaryote. Now, a prokaryote doesn't have many genes, This little loop of DNA is very tiny compared to a eukaryotic organism. They may have, 2,000 or 3,000 genes, whereas over here you have 25,000 roughly in a human, and many more in some organisms. Plants have many more genes than humans, or mammals, vertebrates. Plants have, 50,000, 80,000 genes. We have 25,000 roughly. Now, regulation is how do you use these genes? Well, a prokaryote regulates its genes in order to adapt to the environment. Generally, there is such a thing as turning on and off a gene, but there's also producing more of something. So genes are there to make mRNAs, which then make proteins, which change the cell. The cell can change its chemistry and even its shape and even the structures in it by turning on and off genes and by increasing the output of a gene. In other words, increasing the rate at which it makes proteins. So the idea here is that is regulation. And that'll be the first thing we begin to talk about here in a second. But just realize that prokaryotes are usually regulating their genes for simple survival. Usually find something to eat and digest it, and then if something isn't around, if you're not want something at a particular time, you would turn off the genes needed to eat whatever that is. So you're kind of they're going to turn on genes depending on what's available to eat. If it's not available to eat, they're going to turn that gene off. And that gene would be some type of enzyme, some type of protein that's used for digestion. But otherwise, they're going to leave those genes off. Whereas over here, in a eukaryote, it is a completely different situation. Remember, eukaryotes are typically multicellular organisms with many different cell types. So first, before we go on to, we're going to talk primarily about the eukaryotic cells and their different cell types and how you get different cell types. But let's go over first some definitions or show you some of the keywords that we're going to need to know here as we move along. Well, the first one is gene regulation. And so I want to tell you that gene regulation is exactly like what I just said. There are really two types of regulation. Turning genes on and off, on/off, and we will learn how we, as eukaryotes, as humans actually, do that. And then also changing or altering and I'm going to call it the volume, just like a radio in your car. You have an on and off button, and then you have a volume button. Well, altering the volume of what, though? It's not the volume of the sound. It's not how loud or soft it is. It's altering the volume of mRNAs, which means also if you alter the RNAs, it means you alter the amount of protein you make. So in other words, these are the two ways we're going to talk about regulating genes. Sometimes we want to turn them completely off or turn them on, and then maybe only sometimes you turn them on at different times in your life, that sort of thing. And then we also want to alter how much of whatever it is that they produce, whatever protein that they produce, we want to alter how much of that protein is produced. Maybe we want just a little, sometimes we want a lot. And these are the two types of gene regulation that we need to talk about. Now, we're going to talk about these here in a little bit. Oh, I want to add one more thing down here. Transcription factors. That's another term that we're going to need to know. All right, so, but our first one is, these are the two types of gene regulation that we're going to talk about. Now, we're not going to talk about prokaryotes much. We're going to talk about mainly in this one eukaryotes, us, organisms that have multiple cell types. And when I say multiple cell types, I'm talking about here. Here's a plant, and you look at different parts of a plant. If you look at the cells under a microscope, you'll see that there are many different types of cells. How do you get these different types of cells? Well, you get it with this regulation. You turn on certain genes, which means they are producing proteins, but you turn off others. Not every cell uses the same set of genes. Now, every cell has all of the DNA, in other words, has all of the genes, all right, necessary to produce a particular protein, but most of them are turned off in any given cell. There are some genes that are always turned on, but in every cell type. But in general, each cell is going to have a different set of genes turned on and off. And they may overlap a little. You always need to make things like RNA polymerase, so the gene that makes RNA polymerase is always on. But you may not need certain other proteins in your cells. So the whole idea here is, how does the cell determine this? How does this occur? And what's going on here? So the first part here is to explain gene regulation and what's going on and how we do it as mammals. There are other organisms, plants do it a little different, insects do it different, but mammals are generally what I'm going to explain in this video. We're going to explain how they turn genes on and off and go through this process of development and what we're going to call differentiation. So we'll get there. But a plant does this too. You turn on certain genes and you get a certain cell type. You turn off others, and I'll give you a hint that cancer is when you turn on the wrong combination of genes that confuse the cell. So you're supposed to be a liver cell, not a neuron, or you're supposed to be a fat cell and not a skin cell. And there are specific genes in the genome that determine you becoming that cell type. And if you get a few turned on or a couple turned off that shouldn't be turned off, that cell gets confused and it may die, but it also might become cancerous. Cancer is essentially just a confused cell. It's supposed to be looking like one of these. It gets some genes turned off that it needs to have on, and it gets some others turned on that are from a different cell type, and suddenly you've got a really weird cell, and if it starts to grow out of control, we call it cancer. So just to make that clear, that we are specifically in this video talking about eukaryotes and not prokaryotes. So we're talking about gene regulation in eukaryotic organisms. All right, so the first thing we need to look at is we need to review, let me zoom in here a little, on the DNA and how it forms chromosomes. Now realize this is something that is unique to eukaryotes, and this is one of the methods that we use to regulate genes. It's the structure of the chromosome. How is the chromosome structured? Well, right here we have DNA. Now I'm just going to draw it as a straight line. We know that it's a double helix and double-stranded and has A's, C's, G's, etc. on it. But that DNA has to be wound up into a chromosome. And this is just a picture showing you how it does. And the part that we are mainly interested in is this part right here, and I guess maybe this part here too. So these two, we understand DNA, all right, but these two involve something called histone proteins right here and forming of nucleosomes. So here's the DNA, And we already know that proteins can stick to the DNA. RNA polymerase, for instance, can stick to the DNA and start transcription. Now, what we want to talk about are these little proteins here called histone proteins. Now, here they have them as different colors. I'm going to draw them as just little balls. And there are 8 of these little proteins called histone proteins. All right, I'm going to draw them like this. Alright, and they come together and the DNA will wrap around these. I'm going to do my best to draw this. Alright, so there, it has wrapped around this, and we're looking at it from above. Alright, and you can see that the DNA has wrapped around it, so just like this. Now the interesting thing is, if the DNA is stuck to these proteins, it might limit how well a different protein could stick, like RNA polymerase that we learned about in the last exam. So if we are like this, the DNA out here, all right, is exposed, and out here. And being exposed means that RNA polymerase, let's call this RNA polymerase, if you remember, there is something called a TATA box, T-A-T-A, all right, and if it's out here, this has no problem sticking to that. It's basically working just fine. Same over here. If you had a TA over here, okay, no problem. RNA polymerase can stick there. RNA polymerase. So that means you could make your RNA, and then you could go to the ribosome and end up with your protein. Oops, sorry. It's hard to write with this thing. All right, so these are on, and this is what we call 2 genes that would be on. But let's say there's a gene that's in this area, and it's wrapped around the nucleosome. Well, that means if there is a TATA here, it's already stuck to some proteins. It's stuck to these histone proteins. And if RNA polymerase wants to go here, it can't. All right. RNA polymerase wanted to go here. This gene is turned off because it is wrapped around the histones. So here we can say that this one is off. This gene is off, but these genes are on. This one also is on. So okay, so now we understand how to turn a gene on and off. In a eukaryote, in our cells, if we want to turn off a gene, we wrap it around the histones and we turn it off. If we leave it unwrapped, we leave it on. So now we have answered our first question, and we can say we understand what histone proteins are. All right, these are proteins that DNA can wrap around. Oops. Often, not always, often turning them off. All right, now a nucleosome is this piece of DNA and the histones wrapped around it. So in other words, this is a nucleosome. A nucleosome, here they have it, these little balls right here with DNA wrapped around them. You can see each of those are the nucleosomes. All right, and the DNA wraps around them. The pieces in between, like right in here, they would be on, where the pieces that are wrapped around it are off. All right, and this is one nucleosome right here. This is many nucleosomes down here. All right, and that's our idea, and this is how we turn on and off our genes. We wrap them around the histone proteins, forming a nucleosome. All right, so let's see, nucleosomes, regions in the chromosome, I'll just skip that part, regions where DNA is wrapped around nucleosomes. Histones. All right, and so that's a nucleosome. Now, we have to go on and talk a little bit more about this. Let's move on here. Here we show another picture of this with them wrapped around, and so just to keep in mind, this would be off, all right, off. But here, the DNA, or like right here, would be on. This piece in between would be on in here. So this is our kind of our concept of on and off within the chromosome or in the nucleus. Which genes are turned on and off for that particular cell? Well, it has to do with which ones are wrapped up and which ones are not. And we can think of this as maybe these dots are the RNA polymerase. And RNA polymerase is trying to stick here. I don't think that's what they're trying to show here, but you could think it's trying to stick and it can't. Here's one that tried to stick, and maybe this one looks like it might be successful. It's in between, and so think of it that way. But moving on here. So I don't want to go to this yet. Let's see. I need to explain. I want to go back here just a second about the types of cells. So I said that here you have different types of cells. How do we get different types of cells? We all start out with a zygote. Now, a zygote, you have an egg and a sperm, all right, and the sperm fertilizes the egg. So half of the DNA comes from dad, half comes from mom. They merge, all right, and essentially you get what's called the zygote with 2 copies of each chromosome. All right, sorry. Not drawing that very well, but here you had one copy, and one copy. So if you have two types of chromosomes, chromosome 1 and 2, you're going to need 2 copies of them. You get 1 copy from dad of each and 1 copy from mom of each. Combine them, and you have a functioning cell. Now, we have more chromosomes than 2. Plants have dozens. Plants are more complicated genetically in a lot of ways than we are. But I wish I had more time to talk about plants. They're really interesting as far as their genetics and their genes. But the idea here is we have this, and this is called a zygote. And it is a generic cell. It's really the only thing that might actually look like that cell that you see in the textbook where they show you the parts, the organelles and everything. No cell actually really looks like that. Maybe the zygote does, but the others end up looking very different because what have they done? They're not just using the general genes needed to survive. They're using those plus whatever it takes to be the type of cell they need to be. And we need to turn those on. Now, how does that occur? How do you get this process of turning this cell into literally billions of other cells in your body, and how do they know to become one type of cell or another? And that's the key here, and that's why I needed to teach you about this process, because we need to find out how this occurs. So this zygote here starts out as 1 cell, and then of course it splits into 2 while you're in the womb. All right, and then these also split into other cells, like this. And what happens is they have a generic set of genes that are turned on from the first one. But as they get to this process, they start sending out chemical signals to one another. So they're producing proteins and pumping them out and letting the other cells know that they're there. So this cell knows that it has 1 cell on this side, but no cells here. This cell knows it has no cells here, but it has 1 cell here. And these cells here in the middle know that they have cells on either side of them. So by knowing this, these chemical signals are what telling them that, hey, I have chemicals coming here, and this cell is telling me his location. This cell is telling me its location. And that tells them basically, chemically, what genes to turn on and off. In other words, this one knows I have nothing coming, no signal coming from over here, but I am getting a signal coming from what? From the right-hand side, depending on how this video is flipped or not. This would be the right-hand side. And so this cell then knows to turn on a certain set of genes, and maybe it knows to become a long, narrow skin cell. All right. This one said, well, I have some information coming here, and I have information coming here. I know that I need to be a muscle cell, and I look a little different. All right. So a skin cell and a muscle cell. All right. Now this one said, oh, I have muscle cell on this side, and then I have nothing on this side. All right. Maybe I need to be a fat cell. A fat cell is globular. And then this one says, oh, nothing on the outside, I'm going to be a skin cell again. And so I'm simplifying this completely. But essentially what's happening here, these cells are sending each other information about what's around them, and they're becoming a particular cell type. And what they're doing is exactly what we talked about here. They are essentially unwrapping or wrapping the DNA around the nucleosome. So you can imagine that when you start out, most of them are off, and then this process we're going to call development, they are unwrapping, all right, the DNA and turning on these genes to become certain cell types. Now this is a process, development, all right, this is a process where cells, typically in the womb, all right, let's make that clear when this happens. All right, turn on or off specific genes that determine their cell type. All right, so this is usually permanent, and so once they've decided to be a skin cell, then when they divide again, they're going to make all skin cells. And this occurs before birth, all right, for the most part. Not always, but for the most part. Now, differentiation is the process. This is the process, all right, of cells, all right, actually becoming a particular cell type. So in other words, this is where the actual process of turning the genes on and off. And we'll talk about how that happens. All right, now, so this is this is the process that all cells go about in the womb, all right? So you have to do differentiation to undergo development. In other words, development is the process of all these cells doing it together, all right? This one is the individual cell. This is what an individual cell does. This is the process that goes on inside the entire body, all right? So each cell does differentiation, and in the process of development, this is what the result is. So this is about all cells. This is about a cell. Maybe I should, a cell. This is a process for a cell. That's a better way to explain it. To become to become a cell type. All right, and I'll put this within a cell. All right, So, understand the differentiation is what a cell will undergo, and when all cells are doing it at the same time, when you're developing in the womb, we call that development. All right? Now, we need to talk about how we decide that. What is it that decides this process? Well, these chemicals going in between it are the key. The chemicals enter the cell, and they do certain things, and we need to talk about that. We need to talk about this process called methylation. So we know what a gene kind of looks like now. We know that a gene has a reading frame, and on one end we have the terminator, which basically tells the RNA polymerase where to fall off, and we also have the promoter, the TATA box, and this is the place that RNA polymerase likes to stick, and this piece will be an RNA. This protein will attach here, RNA polymerase, move across here and fall off, but as it moves across, it's going to produce this RNA, which is going to eventually go to a ribosome and make a protein. So ultimately, we make a protein here. Just to review you. How do we turn this off? Well, we wrap it around the histone proteins. And what determines whether or not it gets wrapped around or not? Well, that is determined by something called a CPG island. And that is just a bunch of C's and G's that typically come in front of the promoter, the TATA. So somewhere out here nearby, you're going to have a bunch of C's and G's. And what happens is, if these are not altered, they don't wrap around the histone. But there are proteins that can come along, all right, that can do something called methylation to them. And we draw this wherever there's a C and a G, which is a very rare sequence. You don't see a lot of C's next to G's, except for in front of genes that need to be turned on and off. And we put these little lollipop sticks on them. And if they have the lollipop sticks and they're clear, then they're not methylated. And methylation is just adding a CH3, a carbon and 3 hydrogens onto the C. And if they get methylated, though, where you add the CH3, we color them in. So they color in the lollipops, and they are now methylated. So these two are methylated. This one is unmethylated. So methylated. All right. Right here. Okay. So if this one is methylated, it makes it sticky to the histones. That's the way we can think of it. I am simplifying a great deal, but the idea is it gets sticky and therefore it gets methylated. Now, how does that happen? Well, a protein comes along and methylates it. So think of it this way: when we were thinking about our cells that were talking to one another. So depending on what's going on, the signals coming from this cell tell this cell to methylate certain genes. In fact, the chemicals themselves, the messages can enter this cell and essentially go and do the methylation. And the ones coming from this cell can enter the methylation. So depending on what they are sending one another, they determine what this cell becomes. So the cells around them are essentially sending them chemicals that are doing this to them. And this happens when you're in the womb. So these cells that are here, where's our picture I want? We're back here. So these cells that are dividing are constantly communicating and sending signals to one another. They're sending chemicals that aren't just signals. They're actually the signals, the chemicals that are doing the methylation a lot of the time. Now, sometimes they're hormones, and hormones will also trigger methylation and can turn genes on and off, like when you get puberty. And so these signals, these chemical messages are causing the methylation to occur, which are causing the genes to turn on, or there are signals that say unmethylate it and turn genes on. So a lot of the signals either methylate or demethylate or unmethylate a region, turning a gene on and off. Now, CPGs are very rare. and they don't occur in large numbers except for in front of these genes. So the other parts of the DNA, or any gene that doesn't have CPGs, are probably not going to stick to the histones, and so therefore they have no chance of being turned off. Only the ones with these C's and G's are going to be wrapped around the histones and have the opportunity to be either turned on or turned off or the opportunity of being turned off. Technically, when you are born, most of them are turned off. So actually, in development, I should phrase this so that in development, this process is usually turning genes on, because you start off with most of them off. But in puberty, you also turn genes on, and others later in life you might turn some off, like the enzyme for digesting milk. You will turn that off. A lot of people turn that off later in life after you reach puberty. If you don't get it turned off, you get to eat cheese and stuff the rest of your life. But then if it gets turned off, and sometimes it'll get turned off much later in life. My mother didn't lose that gene until she was in her 60s. I haven't lost it yet, but the majority of the people in the world actually cannot digest milk past puberty. People don't realize that. People in India and people in Northern Europe have the gene stay on most of their lives. But in the rest of the world, it usually gets turned off, and they can't drink milk without getting an upset stomach later in life. All right, so now these things that actually cause the development are also called pioneer factors. We're not going to call them. We call them transcription factors, but this is just all that it's showing you here, is that you can turn genes on and off depending on the chemicals that come into your body. All right, now this is actually what's going on a lot. We're moving on to a different type of concept here. Let me go back to these. So we now know what CPG islands are. They are just C's followed by G's. The P means covalent bond instead of hydrogen bond. So remember that DNA, just real quick here, I have a C here. Well, I could have a G here. That's a CPG. But I could, of course, have a G over here, because C's the other side. That's a hydrogen bond. So that is not a CPG. The CPG is just representing that it's a C and a G on the same strand. All right. So we don't want that. Where was I? I want to talk about these. So a CPG island is just a C followed by G, or I should say clusters of CG near a gene, or before a gene to be specific. All right. And methylation, the addition of a methyl group, which is just chemical. We learned of it. It's a CH3. Carbon and 3 hydrogens. If that's a small subscript, you should make that a little 3, like carbon and 3 hydrogens. All right, so the addition of a methyl group to a C, a cytosine, in a CPG. All right, and that just means a CG, with this being a covalent bond in between them. So we've completed the idea of how we turn genes on and off, how we do development, but how do we control the rate? Now, remember when I started out, I said that this was about regulation, and there were 2 types of regulation. All right, let's see here. Turning them on and off, which we now know, but also altering the volume. So just like a radio, you can alter the volume of how much protein you're making. The gene may be on, but how much are you making? And that's when we come into this thing called transcription factors. Transcription factors are also out front near your CPG islands. So let's do this. We know this guy. We know that's a TATA up here in the front. And we now know that you've got C's and G's out here somewhere. But you might also have some other sequences up front. Now I'm going to just pick one. I know one called GAGA. And this is a transcription factor binding site. This is a piece of DNA that, just like TATA has RNA polymerase that can stick, RNA polymerase, there are other proteins that can stick to different places in this promoter region. All right, so we have another protein, and I believe the one that sticks to the GAGA sequence, so that's TATA box. So this is not called a GAGA box or anything like that. It's just called a transcription factor binding site. And this protein here is a transcription factor. It's a type of protein that will stick in this region to some sequence, in this case GAGA. I believe this one is called GRAPH. I think that's the name of this one, but there are dozens of them. And when it sticks here, now it doesn't do the transcription like the RNA polymerase. What it does is it either helps or hinders the RNA polymerase. And it's really complicated. You can read about it in the book. The DNA tends to bend over and do all of these things. And these transcription factors, depending on which proteins bind here, all right, these will either speed up. So let's say if you have one particular transcription factor, maybe it speeds this up. So instead of making 2 proteins a minute, you make 10 proteins a minute. But then maybe you have another one that sticks and it reduces it down to 1 protein a minute. So you probably usually don't turn it off completely, but you can definitely change the rate at which they're made. And that's what the transcription factors do. So the transcription factors change the rate. All right? These proteins stick to sequences in the promoter region, all right, and control the rate of transcription. So how many RNAs are made? Transcription. And of course, if you control the number of RNAs, you're going to control the number of proteins. All right, and so they can increase the rate, they can decrease the rate. Now, the key point here to remember is that they do this in response to the environment. So usually something is going on and something changes in the environment. So your body, as a reaction, produces this GRAPH protein here, and it sticks here. And so you increase the amount of whatever this is. And that might be something you put into your sweat. So you're hot, and heat causes your body to produce this particular, your cells to produce GRAPH. GRAPH binds here, and it increases the amount of this, and your cells secrete this as some protein in your sweat. All right, so that would be an example of this process. Now, there is one other thing that we need to talk about, and that is for another video, but realize that prokaryotes do this as well. They also regulate their genes, but they regulate their genes in groups. So whereas we have one gene, one reading frame right here that makes one protein, and we regulate each of them individually, what we're going to learn when we talk about bacteria in a different video, we're going to learn that they regulate these in clusters or groups of genes. So each one of these is going to produce a different RNA. Well, actually it's one RNA, but we're going to learn that it makes several proteins, 3 different proteins, and we're going to regulate them all at the same time. And this is actually very efficient. Basically, when you have 3 genes that need to be activated at the same time, they operate together, they never operate independently, then why regulate them individually when you could regulate them all together? And so the bacteria is much simpler, and it doesn't need as much regulation because it doesn't have all these different cell types, so it can get away with something like this. Whereas with us, we do the more complicated thing here and regulate, one gene at a time. We turn them on and off. Generally, the bacteria will do something much simpler, not necessarily easy to understand, but it is simpler than what we do. All right, now that's it for this one. Be ready for one. I have these on the Lacoperon already made, but I thought I needed to make a special one for eukaryotes here.